What does a partial response mean for a patient with terminal cancer? The one I'm thinking of had a very large malignant pleural effusion, fluid around their lungs. They couldn't breathe, right? You could look at the radiology and say this person, they had to have it drained regularly, almost weekly. As they started having it, their partial response, you saw the fluid start going away and be almost gone, and eventually the drain came out. We made a difference.
Welcome to Leading Beyond the Lab. In today's show, I'm joined by Bhaskar Srivastava. Bhaskar has a wide-ranging experience covering all things dermatology, an esteemed academic, moving into the world of big pharma with the likes of J&J, and then into the biotech world with Nimbus, and now Triveni, where you are Chief Medical Officer.
My first day on the job was a meeting with an investor, and I said, okay, what's this about? And they said, we're always raising, right? This expression we have in biotech.
Yeah.
So putting forward a convincing plan, budget it out, socialize it with investors and our other stakeholders, and successfully raise a Series B. We're one of the few who are looking to drug the barrier. Our team has found, we call it a needle in a haystack antibody. And the story just makes sense for eosinophilic esophagitis, for asthma. There's a bunch of itchy skin diseases that people don't understand, and we think the kallikreins might be involved. So it really might be unlocking a fundamental pathway in disease biology.
Welcome to Leading Beyond the Lab. In today's show, I'm super pleased and excited to be joined by Bhaskar Srivastava. Thank you so much for joining us today.
Glad to be here, Lawrence.
Bhaskar has a wide-ranging experience covering all things dermatology, an esteemed academic, a lead dermatologist, working at academic institutions like Harvard, Pennsylvania, moving into the world of big pharma with the likes of J&J, and then into the biotech world with Nimbus, and now Triveni, where you are Chief Medical Officer. Thank you so much for joining us today.
Thank you.
I'd love to hear in your own words a bit of an explanation to your journey and what's led you to your current position.
Awesome. So I'm a physician scientist. I'm a dermatologist and immunologist by training, and I started. I was an undergrad at Harvard, and then I went over to University of Pennsylvania for my MD and PhD. As I mentioned, my PhD was in immunology, sort of basic B-cell biology, but immunology has definitely always been one of my passions and loves, even for my undergrad course. I elected to do clinical training in dermatology, so I went over to Yale for that. I picked dermatology because there's a lot of immunology in dermatology, and also because you can sort of see the organ, the skin, and take samples in a relatively non-invasive way and really understand some of the diseases and some of the pathophysiology. So I finished up all that training in 2011, at which time I started a postdoc. And I think that's when I started asking myself, what's the right balance of clinical care and science for me? I really enjoyed seeing patients, especially the complex patients. I also loved being in lab, but I would say trying to be a physician scientist and split my time between the lab and clinic did not feel ideal. And so I wasn't having fun with either part of them. And I think it's important to have fun rather than,
Because of the time.
Because of the time constraints.
Yeah.
Patient care is time intensive. Lab is time intensive. And I would say considerations are changing over time. How hard is it to get, funding? How hard, how long, how much time do you have to see each patient? Those all felt shrinking a bit. And so I elected actually to veer more clinical initially, and was in full-time clinical practice for a year in various settings, including academia, including a federally qualified health center, which is a really wonderful experience, and ultimately private practice. And I saw a lot of patients, and that was a really important part of my journey for me. I did find myself missing some of the more basic science, the immunology, and the ability to think about new therapies. And I can certainly go more into that. There were certain patients in particular that made me understand the impact that new medicines have, and wanted to be part of that. So in 2016, I went over to J&J. I was in medical affairs initially. I was the clinical lead for the launch of Tremfya in psoriasis, their IL-23 inhibitor, as well as the head of a registry called SOLAR, where they collected information about patients in the real world. I did that for 2 years and then headed over to early clinical development. Also a wonderful experience where I got to participate in a lot of strategy because they were really realigning their immunology and dermatology strategy, as well as the basics of clinical development and leading a team of physician scientists who executed on the programs. I found myself really loving the small teams and thinking, I had also gotten a lot of experience to the business of pharma through these thinking about the portfolio, as I mentioned, and actually meeting with a lot of biotechs as we looked at new and emerging science and assets. And so this is around 2021. I find myself drawn to the biotech. And my wife said, well, if you're going to jump, make sure it's a good one. So I said, well, no one really knows for sure, but I had the privilege of getting an opportunity at Nimbus Therapeutics. And I knew the target, TYK2. It was kind of a hot target in immunology at the time. I knew of the company, their reputation for designing great molecules. And when I met the team, I was told, I said, this is the one that we jumped for. And so we moved from Philadelphia to Boston into the biotech world at Nimbus, where I led the strategy and development of the TYK2 inhibitor that's now called zasocitinib, is owned by Takeda. And so I was at Nimbus from 2021 to 2024. Actually, after the TYK2 inhibitor was transitioned over, I worked on an immuno-oncology program for a year, which is really a growing and humbling experience in many ways. And I learned a lot.
That's completely different, right?
Completely different. Still immunology, but completely different. And we can talk about that as well. As I mentioned, I learned a lot, and you see pivoting to a completely different area, the growth that comes with that, the sometimes discomfort and understanding how to manage there and deliver for patients. In 2024,
which is just before going on to 2024, when you were at, I know with Nimbus, they were one of the companies that were at the forefront of employing concepts and things like computational biology and really taking hold of those things. And why did they do that, and what did you find was interesting about that?
Yeah, Nimbus had been in that business since 2009. The company's been around for 15, 16 years, and I know that they employed a range of methods, proprietary and non-proprietary, in computational drug design to make really beautiful molecules. Nimbus makes small molecules, discovers small molecules, and the trick, kinases are also a major part of their portfolio historically and currently. And the trick is to make small molecules that are very selective so that you can impact the target you intend to impact without having off-target effects. And that can be very challenging for kinases or for other types of targets as well. So Nimbus used the full range of methods at their disposal to design a very selective TYK2, tyrosine kinase 2 inhibitor. TYK2 is a member of the JAK family, and there are inhibitors of JAK 1, 2, 3 that have been around for longer and have some safety liability. So in order to drug TYK2 without hitting JAK1, you have to do some serious computational biology and then bring that into the real world. So they use the theory, but then we often said DMTA, design, make, test, and analyze, where you design the molecule, then you bring it into the lab and really keep your standards very high and say, I'm getting the pharmacology I want. Moreover, beyond that, the molecules I bring forward have good drug-like characteristics, meaning their half-life is adequate. I don't have to dose this 3 times a day. I'm not predicting drug-drug interactions that might limit me, at least not up front. You don't know till you get into the clinic and do more tests. But so I would say that Nimbus and Triveni have had very high standards for the molecules they bring forward. So what does that mean for me as a drug developer? It means that I'm confident that we're going to hit the pharmacology we intend to hit, and that when the team assesses efficacy in a certain indication, you feel really confident that you can do the right experiment. You can dose as high as you need to. So for example, with the TYK2 inhibitor, we were able to take the dose much higher than some competitors because we were only hitting TYK2 and not hitting JAK1.
So you effectively model it out?
Yes. It was modeled out, verified, and then confirmed in clinic. We also made a pretty serious investment translationally, now this is in human beings, to make sure that the pharmacology that we were predicting was what we were seeing. Yeah. And actually with that immuno-oncology molecule I mentioned, and that's HPK1, hematopoietic progenitor kinase 1, that's a target that naturally inhibits T cells. And so it's a double negative: inhibit an inhibitory signal and unleash the potential of T cells to eradicate cancer. And that's what we saw was happening. Now, selectivity in that case means not hitting related family members that have the opposite effect. So certain kinases inhibit T cell activation, most of them actually promote it. And so you can imagine if you don't have a selective molecule, you're not going to get the biological effect you want. You're shooting yourself in the foot. So those were just two of the many, many molecules I worked at Nimbus that, that Nimbus developed, the two that I worked on. And the reason that the computational methods, as well as just the very high standards that the team has, make a difference for clinical trials.
And when did you decide that the time was now to move to Triveni?
I think it was, the TYK2 journey was complete.
When you say complete, what do you mean?
Fully transitioned to Takeda.
Okay.
Yeah.
Okay. Yeah.
And
did they acquire the company or the asset?
The asset.
Yeah,
Yes.
I thought they still existed. Yeah.
Yes. And, HPK1, the immuno-oncology asset, we had brought it to a certain point, and we'd also brought in a leader in oncology. So for me, and we had a few assets at Nimbus. It was and is an amazing company, but the immunology assets were a little behind at that point. And so for me, it was the next step in my career to be a chief medical officer. I was SVP of clinical development there. And also to get back towards a little bit home base for me, which is immunology and inflammation.
That's where the heart is.
It is.
And then, so talk me through your first CMO role.
Yeah, so I was really, I would say, privileged to join Triveni at the time when I did. The scientific premise at Triveni is to drug extracellular proteases with antibodies. When I heard that, it didn't immediately resonate with me what that means, but proteases are basically enzymes that chew up other proteins, and they have to be highly regulated. Your body needs them, for example, trypsin for digestion, but you have to keep the trypsin inactive until it gets to the place where it needs to go, or it could digest your pancreas or, the steps along the way. Now, in the skin, there are these proteases called kallikreins, and 5 and 7 are highly expressed in the skin. And so this is how I got to know the company. The targets are kallikreins 5 and 7, and these proteases, when dysregulated, can cause a lot of damage. And there's a genetic signal in a disease called Netherton syndrome that's characterized by atopic dermatitis, like severe atopic dermatitis, like inflammation and other types of inflammation in the esophagus and the respiratory tract. So I got to know Triveni as a company that had an antibody that inhibited both kallikreins 5 and 7 with strong genetic validation in Netherton's, but also in atopic dermatitis, a much wider indication, but having related inflammation, and a really interesting portfolio that included a bispecific that inhibited these new targets, kallikreins 5 and 7 for atopic dermatitis, but also the second arm, an older target, IL-13. And so I find myself thinking, even before I knew the team, what a great target. Sounds like an amazing molecule. And what an interesting strategy to already put forth something new and have already thought of combining it with something established. So to really accelerate drug development. When I met the team, similar to when I met the team at Nimbus, I was sold. These are, and unfortunately they were sold on me. So, this is a team that speaks their mind, thinks boldly, but also thinks in a strategic, business-minded way, because we need to do that to actually bring drugs to market. So I came in and I knew that my job, one, was to do the entire strategy up front. We were going into a raise. I did not know that taking the job, but we raised our Series B shortly after I joined. And so we had to have all of the assumptions about...
Were you involved? Or was that?
Yeah, I was my first day on the job was a meeting with an investor, and I said, okay, what's this about? And they said, we're always raising, right? This expression we have in biotech,
yeah.
We always be raising in other fields too, I'm sure.
I heard the best time to raise is straight after the last one. Because the
Basically,
momentum is with you.
Exactly. So that was literally my first day on the job, and then over the next few weeks, it became clear that that we were headed into a Series B, and So putting forward the plan of a more granular clinical development plan than we had at that point, to put forth a convincing plan, budget it out, socialize it with investors and our other stakeholders, and successfully raise a Series B. So that was a great way to start. While we're doing that, we're also trying to put our key hires in place for development, because we have trials to start. It's public that we started our first asset in clinic in May in a healthy volunteer trial. So we're really excited about that. In order to start a trial in May, you can imagine you really have to be pretty laced up by late 2024. We had to really know what we were doing while hiring the team across clinical operations, program management, clinical development, biostatistics, regulatory, all the functions that you need to enter clinic.
And as someone who's gone through the various channels with Big Pharma, with J&J, to kind of large biotech like Nimbus and now to Triveni, what do you think are the consistent hallmarks of a great drug developer?
Yeah, great question. I think the expertise have to be there, which generally I feel like in every company I've been at, it has.
Yeah.
The culture is really, really important. You have to make sure that it's inclusive. Drug development is a team sport. So you have to have the right voices at the table, and the teams have to feel comfortable speaking their minds. Some would call that psychological safety. And I really do believe in that. So I think,
as the leader, you have to create that framework and environment.
You have to create that, absolutely. It's a conscious, continuous decision, almost to have a clear vision of the culture and maintain that. Cultures also change over time. They have to. And that has to do with the size of the team, the scope, the type of technology. Drug development used to be, from idea to drug might be decades. Now it might be in indications that are faster, 3 to 5 years. Typically not that fast, but if you're in a rare disease or, certain cancers with high unmet need, it can go really fast. So you do have to adjust your culture, but I think the North Star is always bring the right people to the table, ensure that the culture permits them to speak their minds, to challenge each other in a healthy, productive way. That's how you deliver for patients.
So that's the environment for that high performance. And what about from an individual trait? So say, for example, if there's a physician who's considering going into pharma and following your footsteps, if you like, it's not for everyone. So what are the traits of someone that it might be more ready for?
Yeah, that's a great question. I think the first question is to ask yourself, when do I feel most gratified? Is it when I'm making an individual decision, or is it when I'm part of a team coming and moving forward? Is it when I'm giving a talk as the most important voice in the room, if we're thinking of an academic physician?
Yes, yeah.
Right, or is it when a project I worked on comes to fruition and that project gets the glory, but not necessarily me? And I think either is fine. As I progressed through my academic career, I was lucky to have moments. For example, I got to give a Keystone talk as a graduate student, which is really a privilege, right? It went well. To me, bringing forth a drug and being a middle author on that paper is actually the more gratifying piece than being the lead author on a paper, the presenter at a conference. So I think understanding that type of motivation, I think, is a pretty fundamental split between at least the way it used to be in academic medicine and the way that it is in pharma. So having sort of done that, maybe soul searching and saying, what I want is to be part of a big team and to try to bring forth something brand new that could be a drug someday. You have to listen when you join pharma or biotech. Job number one is, when I started J&J, I would routinely ask respectfully, help me understand what this meeting is about. Help me understand what your function is about and what ways in which I can partner with you. And that wasn't immediate. There's times where I came in saying, well, I know this and this is what we should do. And fortunately, I got feedback that's not how to be most productive.
Yeah, that's the humbling experience.
It is a humbling experience. I think you also have to realize that as a physician, your voice will be, it's a pretty significant voice. People will listen. And so it's best not to do a lot of thinking out loud, if you will. It's best to listen because people will sometimes act upon what you say. And so it's important to really take a step back, listen, think carefully, meet with your team, offline, and then get to a really thoughtful, careful place.
Yeah.
It's different than clinical decision making. You don't often have that luxury in clinic. You do when you're in the pharmaceutical and biotech world too.
There's other pull factors, isn't there?
What's that?
There's other push and pull factors in a biotech or pharma setting.
There are. And the more that you know and the more that you understand, the greater your ability to come up with options, right? Clinical development is not done one way. There are options, and those options incorporate scientific considerations, regulatory considerations, business considerations. You have to really understand how you can bring forth the value for patients.
So we're bringing it back to the question there then, Bhaskar. Is it a case of, if you're driven by innovation, something new, very much growth mindset, and you're comfortable and excited by the kind of commercial aspects of taking a drug to market, then it's probably worthwhile considering. Likewise, if you want to be entrenched in the science and are fully focused on being a single voice, then it's worthwhile double checking if it is.
That's fair.
Yeah?
Yeah.
Yeah, it's interesting, isn't it? Because I think the people that I speak with, they always have an intrinsic motivation to help the patient and benefit the caregivers, and that's always the key driver, and that's a key consistent thing for all of the drug developers in the industry that I've worked with.
Yeah.
The ones that move to biotech, there's different things. I find that the ones that are suitable or suited or enjoy and are passionate about staying in the big pharma setting, they tend to have different motivating points or things, but they're consistent with each other. Likewise, in the biotech side, there's this entrepreneurial itch they want to scratch. There's a kind of a dynamism that they just want to embrace.
Yes, I think entrepreneurial is the key word there.
Yeah.
And it is quite possible and desirable to be entrepreneurial in big pharma, and that is at J&J we use that word, and it was in the culture that when you're thinking of your program, you should take an entrepreneurial approach to it, right? But you are embedded in a very large portfolio, right? And considerations and decision making are going to be at that level. In biotech, typically, right, your portfolio isn't quite so large, and you're a small team focused on one or a small set of assets, and you're just fully in, and you have to think like every day really matters. It matters in big pharma as well. In biotech, you feel that somewhat more keenly. You also will likely be wearing more than one hat in biotech, almost surely. Especially at the beginning, when you move in as a CMO, you're probably going to be wearing all of them in development, because part of your job is to hire the team. And so typically most of those folks will not be in place. So I think that those are some of the attractive or distinguishing features of biotech. And so if you're a physician in pharma, to ask yourself if you want to jump to biotech, you have to put on those considerations of how much risk do I enjoy, how much do I enjoy wearing multiple hats.
And from a leadership perspective, something that's quite telling is that you have to manage teams in different environments. What are your go-tos when it comes to building and leading that team? For
me, it's changed over time, right? Because the types of teams I've hired have changed over time. So
what do you mean by that?
So let's take when I was a senior director, hiring physicians to execute on trials is different than building an entire development organization. So going back to when I was in that role as senior director, saying hire director, associate and director level physicians to execute on the trials, a huge part of it was making sure that those folks it's somewhat narrow. You're coming in, sometimes from academia, sometimes with a few years of experience in pharma. How comfortable are you leading trials and thinking in that way? So there'll be a lot more of the technical aspects to dive down on there, and also setting the right culture. But at that point, I'm really leading a function. Now, moving forward to the way I hire teams today, it's across expertise and functions, many of which I do not know well.
An example?
Pretty much anything other than clinical development, right? Would I call myself someone who fully understands what clinical operations does on a day-to-day? No, I haven't done it. Do I fully understand everything about regulatory, about biostats? No. So I understand enough to assess some degree the technical skill. I have to be able to ask the questions that make me comfortable with that. And then it becomes my job to put the right experts together, set a culture, and to some extent get out of their way, but check in enough. So it's a real balance, I think, of how do you build an entrepreneurial team with a high degree of psychological safety, still maintain the accountability and make sure, because you as a CMO are accountable for that at the end of the day. So that becomes the art of leadership as you get further and further away from the subject matter that you're comfortable with.
I'm surprised how frequently getting out of their way has come up in the last 3 episodes of this podcast.
Yeah.
Because I think maybe it's because we're gearing specific leadership type questions, but you don't know what you don't know. So creating that framework for the psychological safety, as you so eloquently put it, and then giving the experts their opportunity to shine, I suppose is a big part of leadership, isn't it?
Yes, it is.
Yeah. And so from those different environments, are there any key milestones or is there one specific achievement where you think, do you know what, that's why I do what I do. That's why I come into work.
There have been several. I've been lucky in drug development. There have been successes that I've got involved with. TYK2 at Nimbus is one of them.
Yeah.
It was a small team that delivered great data and a lot of data. And I would say a phase 3 ready asset in many ways, right? If you look at the time between when the transaction happened and when the trials actually started, Takeda, there was very little gap.
Yeah.
And so very proud that a small team led, multiple phase 2B trials, multiple phase 1 trials, but also built the value in the CMC package, right? You have to make enough of the drug. You have to encapsulate it in certain ways to do trials. The translational package, the toxicology package, you want to deliver for patients. In order to do that, you have to stay on your timeline in your value creation. So it was a biotech experience, but very much created a drug that was ready to go as fast as a bigger organization could take it. I'm very proud of HPK1. That drug is still being developed, and immuno-oncology is challenging right now in terms of bringing assets forward because there have been a lot of blips, let's say, with different approaches since the days of PD-1, PD-L1, but we made a difference for patients. This is public. We've shown posters where we had complete responses, partial responses in patients who had tried everything else. And what does a partial response mean for a patient with terminal cancer? The one I'm thinking of had a very large malignant pleural effusion, fluid around their lungs. They couldn't breathe. Right? You could look at the radiology and say this person, they had to have it drained regularly, almost weekly. When they had their partial response, as they started having it, you saw the fluid start going away and be almost gone, and eventually the drain came out. So regardless of the ultimate fate of that asset and whatever the business considerations are, et cetera, we made a difference for patients. And that, if you ask me to weigh that versus TYK2, I can't, right? They're equally meaningful to me.
One thing that resonated that you said earlier in this conversation is that on your first day you were talking to the investors. So a first-time CMO, obviously with an incredible background, but still it's your first role as a chief medical officer. It's a different type of conversation. So how did you go about that conversation? How did you prepare yourself to have a conversation with a completely different type of stakeholder?
Yeah. So I was a little bit prepared because at Nimbus, and this has happened to me a few times in my career, the CMO who hired me left for other opportunities a few months later, and they didn't replace her, at least not immediately. We had a CMO coming towards the end of 2022. So
Enter Bhaskar.
What's that?
Like Enter Bhaskar
right.
Training.
So as I was SVP clinical development, but I also got a lot of stretch opportunities. And as I mentioned, that's happened to me a few times, which is helped me grow, actually.
Yeah, I was thinking what you're saying.
That happened actually at my first job at J&J, where the person who hired me left to be in global med affairs. And so regardless, I came in with some experience we had been through fundraising at Nimbus, and as well as, diligence, as we call it, the process of leading up to selling or buying a drug. So that was good. But I will say it's different when you're CMO, right? For real,
because the buck stops with you.
The buck stops with you.
Yeah.
At Nimbus, I was, our CEO was the one who led a lot of the conversations with support from me. In this case, the investors were looking at me. What's your development plan? So I think you have to know what to say. You don't want to go too far. So you have to have enough detail that it's believable. But you also have to sort of leave certain things open, make sure you're reading the room, get the reactions. It's harder virtually, right? I will say it is easier in person, having done both types of interactions now. So there's a lot of judgment that goes into it, and you're definitely going to hit situations where you don't have the answer. So how do you show up when that happens? I think is a key part of being a leader.
How do you?
You say what you know, you're clear about what you don't know, and then you follow up on it. Yeah.
So it becomes like a developmental point.
It does, if
Yeah.
those come up. Now, ideally, not too many of those should come up, right? I was hired to be CMO in an area that I'm quite familiar with, so it wasn't a lot of that. But you don't want to be too specific and then end up not doing that as your plan. So
one of the feedbacks that we get from investors specifically is that the pitch or the interaction, sometimes technical people can go into too much detail almost.
Yeah.
And being able to simplify that message and the value proposition is a key part.
Yes.
I'm interested to see if that was something that kind of flagged in your brain, or if you had to do that, or if you naturally did it just because you were reading the room, like you said earlier.
Yeah, you do have to pivot a fair bit in how you explain something according to your audience. So that is a skill that I think should be cultivated. Different people have different tendencies to do this anyway. I think it's good to be a good storyteller, and it's good to practice that. I think in the day-to-day, one should practice telling stories, adjust it to your audience, and see what resonates. In a given day, I might be interacting with a very technical audience, asking me about the pharmacokinetics of our drug and how that plays in, and maybe I don't have my pharmacokineticist in the room, my clinical pharmacologist, so I'm going to give the best answer I can. Or if I'm at a different audience with investors, I may, instead of going into technical details, say we're able to achieve a high enough level of this drug to really inhibit our target and make a difference for patients, right? That's very different than the Cmax is 100-fold over, the expected minimal efficacy. They're kind of saying the same thing. So you just have to use different words and different phrases and rely on different comparisons to make impact.
So one is more kind of generally impact related. Yeah, impact, that's the right word, isn't it? Impact related. And through that journey, is there any specific lessons that you just think, I wish I could have told myself this on my first day at J&J, for example?
Absolutely. I think going into different conversations, it's important to align with your own team. If you're a CMO, then with your C-suite executives especially, about the intent and the tone. There was definitely an example I can think of where I went into a conversation with an investor with a more conservative tone than was warranted at that point. A flip had happened, we had gotten some data, and that was the time to shift from discussing caveats to, hey, we've got this and we've got it contextualized. And again, in a busy day where you're having, half an hour meetings, 20 conversations a day, and you're shifting gears, it's very easy to not prepare yourself in that way. But so the leadership team and I at Triveni take time to discuss that with each other. Hey, we're having this conversation tomorrow. We meet Monday morning and talk through our conversations and make sure that we're aligned in the level of the pitch and the intent.
Yeah, I think, and in this so-called golden age of dermatology, where do you think the future trends will go?
Great question. So I do think, I find myself wondering, is it new modalities or the old modalities? Small molecules and antibodies against new targets, what's going to make a difference? I think it's a combination therein. I think one has to think about new biology and new ways of making a difference. So let's say for atopic dermatitis, first line of many textbooks about AD is atopic dermatitis is a barrier disease. The major genetic determinant is a barrier component called filaggrin. The therapeutics that we have are largely immune focused, right? They focus on cytokines, cytokine receptors, or the signals that cause T cells to become overactive. So we've drugged the immune system in atopic dermatitis. We haven't directly drugged the barrier. And that's where Triveni's approach is incredibly interesting and alluring to me, because we're directly inhibiting the enzymes, the proteases that damage the skin. And so we're pushing into a new aspect of the biology. It's harder to drug in many ways than immune components, or at least we made less progress, right? An antibody against a protease is a harder antibody to find than one against a cytokine or a cytokine receptor. So I'm really glad that we're doing that. But that's using an established modality, a monoclonal antibody, just developed in a clever way. There's new modalities that we can explore, whether they be gene therapy for certain more rare diseases, whether it be T cells that destroy immune cells, to put it kind of at a high level, but CAR T cells or bispecific T cell engagers or antibodies that bind immune cells and destroy them. I think many of us were surprised wonderfully when the data started coming out that you can do that and make a difference in devastating diseases like systemic sclerosis, where we just didn't have anything before. So I think thinking broadly about the biology, deploying all of the means that we have at our disposal, I think is really important, and not being limited to thinking, well, this path or that path will get us there. I think we need to think broadly.
I really appreciate that answer. And how does that flow into Triveni specifically, and what sets them apart? Because you're someone that has a super desirable profile in a super competitive time within dermatology. So what was it about the company that you think is going to set them apart from other dermatology biotechs?
We're one of the few who are looking to drug the barrier and have, I think, the right approach to it. So, as I said, the immune system has been the focus in a lot of dermatologic diseases. Partially that's because it's very important. We know that. Partially that's because it's where our drugs where we've been able to make drugs, literally where we've been able to drug something. Now, our team has found, we call it a needle in a haystack antibody. It's called TRIV509. It binds kallikreins 5 and 7, and it binds them both with high specificity and selectivity. So we can really go in and start modulating this biology. And we have, as I mentioned, the genetics point us in direction that this should make a difference. We've shown data at the recent Society of Investigative Dermatology meeting that in explant tissue, pieces of punch biopsies from patients with atopic derm, adding our drug to that versus a control drug creates fundamental changes that correlate with the improvement of atopic dermatitis in the way that the skin, normalizes, and the kind of inflammatory and barrier look of the skin, without getting too technical. So I think quite simply the approach, and it's with an antibody, so that's a proven modality. So once you show that you can impact the disease, going from there to
less
drug
risk.
Has less risk. Yes, I'm not going to say none, and knock on wood and those kinds of things. This is drug development, but the modality itself is de-risked. We do have the bispecific that combines kallikrein 5/7 with IL-13. And I think that's very promising. Bispecifics are more risky than monoclonal antibodies, but we're making good progress there. So I think unlocking the potential here, and not only atopic derm, but there's genetic signals in other other barrier diseases, and the story just makes sense for eosinophilic esophagitis, for asthma, for to put it kind of at a high level, there's a bunch of itchy skin diseases that people don't understand what is going on there. And we think the kallikreins might be involved. So it really might be unlocking a fundamental pathway in disease biology, particularly skin biology, but also beyond. Moreover, I think the idea of drugging proteases can push into other organs. So this is beyond skin, but what are the proteases that damage the joints? What are the proteases that damage your gut, other organs? So I think there's a lot of potential there.
And I suppose my final question would be, what is the next milestone or big thing that Triveni is working towards?
So we've started our trials with TRIV509, and so getting patient data in atopic dermatitis is going to be one of our next major milestones. And so we intend to start those trials in atopic dermatitis patients shortly. We will be going for sort of efficacy in the registrational atopic dermatitis population, meaning patients with moderate to severe disease who have all the hallmarks that you would want for that indication. As well, we'll be doing mechanistic studies to really understand the biology of kallikrein 5/7, and we have a lot of great collaborators there. So I would expect, within the next year and a half, a lot of data coming out that tell us exactly what type of a difference we can make.
Wait and see.
Wait and see.
Thank you, Bhaskar. I've really appreciated your company, and thank you so much for giving us your time today.
My pleasure.
Just for the audience members, if anyone wants to reach out to you to find out more about anything that you've spoken about, how would you suggest they do that?
Yeah, please connect with me on LinkedIn. I think that would be the
Great.
straightforward.
Thank you.